Welcome to my video project for Intro to Biochemistry. My name's Ashley, and today I'm going to teach you all about enzymes and how they work, including the lock and key and induced fit models with real-life examples. Have you ever wondered how plants are able to make their own food from sunlight, or how humans break down their food? Well, to put it simply, the answer is enzymes. Enzymes are macromolecules that speed up chemical reactions in living things. Without enzymes, many biological functions would take significantly longer and would make it hard to survive. Enzymes are macromolecules that speed up chemical reactions in living things. Without enzymes, many biological functions would take significantly longer and would make it hard for organisms to survive. Imagine you were on your way to school and you had to ride your bicycle over a large hill. The hill represents activation energy in a chemical reaction, which is how much energy it takes to get over the hill. But you discover a shortcut just beside it, with a shorter hill. At the end of the day, you would start and finish at the same places, but it would take overall less energy to bike up the smaller hill. This is also what's happening in chemical reactions. Chemical reactions require energy to function, and catalysts provide an alternate pathway that is sped up and has lower activation energy. So, what are enzymes? Enzymes are protein biocatalysts that help to speed up biochemical reactions in living organisms. Enzymes are not used up or changed in the process, and their names typically end with the suffix ase, for example, lactase, amylase, and ligase. Structurally, some believe that enzymes resemble a pizza missing a single slice, or even the famous video game character Pac-Man. That little space that appears to be missing on an enzyme is called the active site. This is where the molecule enzymes work on, called substrates, would bind to. Once the enzyme has bound to the active site, the compound is called the enzyme-substrate complex. Originally, scientists believed that enzymes and substrates fit together in what was called the lock and key model, where the two counterparts have a rigid, complementary shape and fit together perfectly. In this analogy, the enzyme is the lock and the substrate is the key. This would also suggest that a specific substrate will only bind to the corresponding enzyme. For example, sucrose, which is a sugar, can only bind to the enzyme sucrase. Remember how I said that enzyme names end with ase? Similarly, the names of sugars typically end in ose. Knowing this will help to distinguish which enzymes break down which specific sugar molecules. However, there is now a much more modern and widely accepted theory. This is called the induced fit model. This theory suggests that enzymes have a more flexible structure, and once the substrate binds to the enzyme, it can slightly alter its shape in order to form to the substrate, like a glove fitting onto your hand. Now that we've discussed the two models, we're going to look at two real-life examples of enzymes that you probably use every day to help break down food. First example we're going to look at is called amylase. Amylase is an enzyme present in your saliva that works to break down starch. Starch is a common macromolecule and can frequently be found in bread. Another example of a digestive enzyme would be lactase. Lactase breaks down lactose, a sugar commonly found in milk, into smaller sugars like glucose. Lactose-intolerant people don't naturally produce as much lactase in their body, which makes it hard for them to digest dairy products and can make them feel sick. You can think of lactase as a pair of scissors that can snip lactose, which is a disaccharide containing two sugar molecules, in half in order to break it down to smaller pieces. Since enzymes are proteins, they have certain temperatures and pH levels at which they work best. In the human body, enzymes are most functional at body temperature and a natural pH level. If the enzyme environment was too acidic or too cold, for example, the enzymes would stop working properly, also known as denaturation. So here's a quick recap on what we learned here today. Enzymes are biological catalysts that speed up chemical reactions and provide an alternate pathway which lowers activation energy. There are two major models of enzymes, the lock and key model, which shows how specific enzymes are, and the induced fit model, which shows enzyme flexibility. We then went over two real-life examples of enzymes, amylase, which breaks down starch, and lactase that breaks down lactose. Ultimately, enzymes are crucial to biological beings, and life would be much slower without them. I hope you learned something new today. Thank you for watching.